everyone. So I'm George Mehta. I'm the Executive Director of Singapore Eye Research Institute, and we're going to just change the order a little bit of the talk. So I was going to go first to give an introduction of SERI, but we'll do that at the end, I think. And then my colleagues from SERI, I have three here: Dan Melia, Rachel, and Katrina Bell. And SERI is a research institute in Singapore. It's one of the largest eye research institutes in the world. And we're going to be talking about trying to do innovation in eye research and why it's important to help us with our clinical practice. So my first speaker today is going to basically be Rachel Chong. She's a young clinician scientist in the glaucoma service at SNEC and also in research in SERI as well. Thanks, Rachel. Can we get Rachel's slides?
Okay, hi everyone. My name is Rachel, and I'm a glaucoma consultant at the Singapore National Eye Centre and also a clinician scientist at the Singapore Eye Research Institute. So thank you very much for the chance to speak to you. I've been asked to share some pearls from my journey as a clinician scientist and some of my experiences thus far. So I do have a presentation. Could we? Yes, thank you very much. Great. Okay. So one of the questions I like to ask myself from time to time is, what drives you? So what makes you excited to be up in the mornings to come to work? And for me, I think the privilege of being a clinician scientist is something that I'm very fortunate to have. And at the very start of my journey, it was actually an elderly lady in England whom I befriended in medical school who taught me everything that I needed to know about glaucoma then. So this is just a stock photo from the internet, but the lady circled in red does bear some resemblance to my friend Dorothy. So she was a lady in her 80s who was very healthy and had a very full life, apart from the fact that she suffered from glaucoma. And I remember her telling me that the vision that she had in one eye was about the size of a dinner plate held in front of her. And she was blind in her other eye from very advanced glaucoma. And at that point, I thought to myself, gosh, I really wish there was some way to help patients like her. And she was really the one who got me started on my journey as a glaucoma specialist and also clinician scientist who was interested primarily in trying to improve the lives of patients who have this disease. So when I was in medical school, I was fortunate also to meet Professor Ang Tin, who is now the head of the Singapore National Eye Centre, whom I did an elective with. And at that point, it was actually a very simple project, basically looking at how pupil dilation affected intraocular pressures in patients with narrow angles. And halfway through my training, he introduced me to Professor Keith Martin, who became my PhD supervisor at the University of Cambridge. And while at the Singapore Eye Research Institute, I've also been very fortunate to have the mentorship of Professor Leopold Schmetterer, who is an ocular imaging expert. And so really throughout the course of my journey, what has been extremely helpful has been to find mentors who are both willing to listen to your ideas, as prematurely formed and as crazy as some of them might be, but who are also very willing to offer honest feedback. And ideally, if you could have access to a few mentors, more than one, that's tremendously helpful as well. So when I embarked on my PhD at the University of Cambridge, I had just finished my basic training as a resident in ophthalmology. And at that point, having cleared my exams, thought that I knew everything that I needed to know at that point. But delving headfirst into a basic science PhD was extremely humbling. And then I realized that actually I knew very little about glaucoma, despite having learned about it from many textbooks. So my interest at the point was really to find out more about the retinal ganglion cell, which is, as you know, the primary cell involved in glaucoma blindness. And one of the first things that I did was to do cell culture that involved growing retinal ganglion cells in a Petri dish. And as you can see here, even though as glaucoma clinicians, the main things that we assess are retinal ganglion cell axons, because these are what we can visualize at the optic nerve head. Actually, the retinal ganglion cell is a very, very beautiful cell. And as you can see here, you have the green branching structures, which are the dendrites, as well as the red dots, which are the synapses. which illustrate where the retinal ganglion cell can form connections with other cell types. And so I was very intrigued by this neuron in the retina. And I went on to culture them and study them. And one of my images actually ended up on the front cover of IOVS, which I was very proud of as a PhD student with very little basic science training at that point. So apart from cell culture, my other experience with glaucoma science during my PhD was also to look at experimental models of glaucoma. So one of the things we used at that point was the laser trabecular meshwork model, where laser was applied to the outflow structures of the drainage angle in rats. And then that caused intraocular pressure to increase, and we were able to study the effects of acute intraocular pressure increases as well as chronic pressure rises in the eye and how that affected the cell types in the retina. So even though this is a model that I don't really use in my current research, it was nice to be exposed to these things because that gave me a much deeper understanding of experimental models of glaucoma in general, as well as the advantages and pitfalls of using some of these models. So at the moment, my work at the Singapore Eye Research Institute has been really to look at intraocular pressure independent glaucoma mechanisms. And part of this has involved looking at the genetic risk factor for caveolin-1, which is a protein that's expressed in something called the neurovascular unit of the eye. And I'm interested now in really looking at how the different cell types in the retina and as well as the optic nerve head talk to each other in glaucoma. So this process is called neurovascular coupling, and it's quite well described in the brain, whereas in the eye, it's essentially a collection of interactions and crosstalk signals between the blood vessels and the glia, as well as retinal ganglion cells, as you can see in the red box. So looking at some of these transgenic mice, they have a very mild intraocular pressure elevation in comparison with the normal wild-type mice. But because caveolin-1 has been implicated as a glaucoma genetic risk factor, for me, this was very interesting to study. And we then went on to look at how this affects the blood vessels around the optic nerve head, because as you know, ocular perfusion has also been implicated in glaucoma pathogenesis. And so just looking at the fundus photos, you can see that it doesn't appear to be any major changes in the caveolin-1 deleted mice. But then when we looked at something called the neurovascular coupling response at optic nerve head, involving basically whether or not there are changes to perfusion at optic nerve head when you shine a light at the eye, we found that in the mice that have got caveolin-1 deleted, there was much less of a response with 3 minutes of a flicker exposure. So in a wild-type mouse, if you shine a light at the eye, the blood vessels around the optic nerve head dilate, and that enhances blood flow in the area. Whereas in the mouse that had caveolin-1 deletion, we didn't see that response. And so that was actually really interesting because then when we looked at the vessel morphology at the superficial as well as the deep capillary layers, there was no difference. And in terms of retinal ganglion cell losses, actually it was very similar in both wild types and caveolin-1 deleted mice. So this is just a brief introduction to what my research entails at the moment, looking at IOP-independent mechanisms in the lab involving animals. And in addition to this, in terms of my clinical research, I'm also running a dedicated service for patients with high myopia and glaucoma. And again, from the perspective of thinking, are there any intraocular pressure independent pathways that underlie the risk of glaucoma, which as we know is also much higher in patients who have high myopia. So towards this end, we're looking a bit more closely at structure function mapping in patients with high myopia, because as you know, the diagnosis of patients of glaucoma in patients with high myopia is extremely challenging, and that arises from both structural as well as functional abnormalities that are often detected in patients with myopia, whether or not they have glaucoma. And so again, just going back to what I was doing previously with my translational model research, the idea is then to feed back into my interest in basic science and think about whether we can use experimental models of myopia to investigate some of these pathways and mechanisms in closer detail. So just to sum up, as I mentioned before, it's important to try and identify mentors if you can to help you along in your journey as a clinician scientist in visual research. This is just some advice from an excellent neuroscientist, the late Ben Barres, who says that you should find someone who's not just an excellent scientist, but also a good mentor. And you can look up this reference in a journal called Neuron if you're interested to find out more about what he says. Yeah, so finally, just a few words of encouragement. I think for all of us who start out thinking about pursuing research as a scientist after being trained as a clinician for many years, it can often be very, very daunting and confusing because, there's so many things that you might be interested in exploring. And yet it's also a little bit confusing because you might not know where to start. But I just wanted to encourage you all that, you just have to put one foot forward and you never know what you might find. You might be bitten by the research bug. Okay, and finally, why do research? First of all, because somebody has to, in order to advance our knowledge of science and push the frontiers of medicine and pursue better healthcare outcomes for our patients. And secondly, I feel what we really have as an advantage is, as clinicians, we can come up with problems or questions that are therapeutically relevant because we do have the privilege of seeing patients on a daily basis in our clinics, and we're very intimately aware of what their needs are, what their concerns might be. And I think we are also very uniquely positioned to understand what these gaps are and think about how best to address them. And finally, I think most people who are interested in science would have the curse of curiosity, really, where if you have a problem, a burning question in your mind, you just cannot help but want to try and answer it every single day of your life. So that's it from me. Thank you very much. That's my email address at the bottom if anybody would like to write to me with any questions. And I hope you enjoy the rest of your meeting. Thank you.
Thanks, Rachel. That's very, very interesting. So it's interesting how you use your clinical research, which is in myopia and glaucoma, and then go back to study that in an animal model from previous work that you did as an animal model during your PhD. So just if you just give one tip, what would you be useful that you wish somebody told you back when you were PhD, that you wish you knew, basically, that you'd learned along your journey?
Yeah, I think an important perspective that I gained over time is just try not to be too pigeonholed and to have an open mind about where your training will lead you. Because I think sometimes it's tempting to think you're only good at one thing or that, your skills aren't actually transferable. But, I think just being prepared to learn new things and always being ready to pursue new skills, even if you know nothing about it, but just having that courage to go and try it has been really rewarding.
That's great. Okay. So we'll go boy, girl, cat. We'll go boy, girl, boy, girl. Okay, thanks, Rachel. Okay, so my pleasure now to invite Dan. Dan is a professor in neuro-ophthalmology. I know they're quite rare around the world, and especially French ones. And so Dan has done a lot of work in AI, and obviously everybody is familiar with AI is really becoming the new fifth revolution now around the world. So he's going to talk about some of his work in using AI in neuro-ophthalmology as well, and how this is has now already and is going to affect our clinical practice. Thanks, Dan.
Thank you very much, John. Thank you very much, everyone, for coming. Can I have my slides, please? Right, so how does this work? There you go. This is my institution, Singapore National Eye Center at the right, Singapore Eye Research Institute. I would like to thank Professor John Mehta for inviting me. I'm very happy to share this session with my colleagues. And it's interesting, I did not discuss with Rachel at all. And interesting, I'm doing pretty much the same subject as you do. So with a couple of different points though. So why do I do research, or why I think it's interesting to do research? A couple of reasons. One of them is just because you're fundamentally curious. You cannot, there are guys who are just learning penicillin is good against infection and they just learn it. And there are other guys who think, why not? Can we find a better medication? You are fundamentally curious. You need to innovate. You need to do one of those guys who, as soon as he gets a new bike as a kid, has to dismantle it. You want to innovate, to do new things, or you want glory. That's another reason. It happens all the time. My grandmother thought that I was the most brilliant ophthalmologist in the world. And I didn't do much to contradict her, I have to say. You want a better career? There are not all the reasons to do research are noble, or you are afraid to get bored. I mean, in this room, everybody's very young, especially my director here, Professor Mehta. But everything is very exciting for you. But with time, things are getting boring because we tend to do more or less the same and same things. Research is a wonderful window to new things and to get intellectually stimulated, at least to me. And after all, you want more joy in your life. And I think research is a wonderful opportunity for that. So I have a couple of suggestions for those of you who are interested in research. In brief, choose your environment, choose your mentor. You heard from Rachel how important it is to be in the right place. You know that you are the son or the daughter more of your environment than of your parents. Choose your target. I'll come back about that. Choose your collaborator. That is very important. Who are you getting in your team when you get older? And choose your values. I'll give a couple of examples, but before, who am I? As Professor Mehta said, I'm a neuro-ophthalmologist at Singapore National Eye Center. I'm dealing with patients with neuro-ophthalmic conditions, but I also, I'm a visual neuroscientist dealing with visual or motor problems. And I was educated in Paris, in France, and the Pitié-Salpêtrière Hospital, which is a quite famous hospital there. I'm talking about environment, and I had a chance to have beautiful mentors, as Rachel had, as Katrina had, I'm sure. And I did a PhD in Collège de France in Paris, a prestigious place, hard work. But I missed something, and I'll show you what it is. One of my research was to look at how the brain controls eye movements. And it is super complex, and this is functional MRI. You're familiar probably with functional MRI. These are neurons at work. This patient, in fact, it's me, and I'm looking right to left, and those are the networks in the brain that are activated during saccades, during horizontal eye movements. And then we wanted to use this as an excuse to understand better consciousness, the neurobiology of consciousness. What does it mean to decide something? And we looked what happens in our brain when I'm deciding to do a saccade before performing it and recording that activity. And these are the images. Look at that. There are even more. That's not only the same networks involved in doing the saccades, but even more, which was a fundamental change, that deciding something means also already executing it, and executing something means just a disinhibition. And I thought this was quite cool, but I don't think I got encouraged enough. We published that in a cool paper journal, Neuro Report, but not a great paper. I'm sorry, you cannot see all the screen if you can adjust it. Not the best journal, because I didn't have the right environment. I don't know why, but I missed that. And in fact, another team just published that exactly the same thing for hand movements a couple of years later in Science. How frustrating. Science. So things have changed since then. I've been in SERI at SNEC in Singapore since 2012, and visual neuroscience. It's interacting with various other fields, but I just wanted to talk about two things that we did over the past 10 years, because the title of this is Science and Innovation. So as an innovation, we created a color pupillometer. It's a small device, it's a new device, it's an invention, looking at the pupil as a biomarker of visual function. And interesting enough, this is the pupillometer. I've had wonderful collaborations with Prof. Tin Aung, you saw him earlier in the slide, and with Raymond Najjar in my lab. And this little device, it's handheld. It provides an objective evaluation, a couple of seconds, and it can be done by the patient himself. But most importantly, it is a wonderful tool to assess glaucoma. And not only look at the area under the curve, wonderful results, pretty much comparable with the OCT and the visual fields in terms of performance. But also, very interestingly, it can assess also it can also classify glaucoma in terms of severity, which I think is very interesting. Again, this is a tool that costs $200, can be held in your hand. And as Professor Mehta said, artificial intelligence is one of the main points in Singapore. And you know probably Daniel Ting and Professor Chen-Yin Wong, who have developed one of the main centers in the world about applying artificial intelligence to diabetic retinopathy, and one of the landmark papers in 2017 about detecting diabetic retinopathy, and how important it is to be in the right place, in the right environment, seeing these people all the time, all day long. I thought, why not just use the same thing? Being in such this is the team in Singapore that was dedicated. It's a couple of years back. People, engineers, ophthalmologists, neuroscientists, neuro-ophthalmologists, all sorts of people passionate about artificial intelligence from various institutions. CERI has been able to crystallize all this talent. And this is my team. And we moved as well into artificial intelligence with the aim to reconnect the eyes with the brain. So the question was, is there a place for AI in neuro-ophthalmology? And I was told, don't even think about it because the numbers are so small. You will never be able to do something. As you probably know, AI is very much data hungry. AI needs big numbers. And neuro-ophthalmology how often do you see those patients with bilateral papillary edema? You are probably experienced and seasoned enough to recognize that. But imagine an emergency room where doctors should see and identify this papillary edema. They are probably not trained enough to do. Why is that important? Because can be hidden behind. You don't want to miss that. And it happens all the time when you see those patients. You don't want to miss them. So the question was, could we teach a machine just to be able to recognize papillary edema in a second like this? Well, the answer was probably not, because we don't have the numbers, but I didn't give up. I just didn't give up. I said, we have to find a way. What was the answer to that? It was friends, collaborators worldwide. So we have created a consortium called BONSAI, like brain and optic nerve study with artificial intelligence. And now we have probably more than 30,000 images. Remember, this is neuro-ophthalmology. This is rare diseases. Yet we have more than 30,000 images now. And we asked in 2018, that 100 years ago in terms of AI, 2018, right? Can papillary edema be diagnosed by a machine? And we worked hard on that. And this is our system. And look how interesting. So this is, we just upload the image. This is a software. And the machine updates to everyone. First year residents will probably be able to diagnose that. That's not a big deal. Everybody can do that. What happens if we are having a more subtle thing? I think that's the interesting. Look at this image now. You know, it's not very clear. It's not beautiful, but can you say this is papillary edema? There are three options. It can be either look at this, the machine. I'm fine. I'm not going to move anymore. I'm not going to move. I'll behave. And so in this case, thank you. And in this case, look at that. The machine is just as confident as in the previous machine. Why? Because the machine saw thousands and thousands of images, right? As a result, so the performance is excellent. And those are heat maps. And we were rewarded because we succeeded to publish this in the New England Journal of Medicine. And it's quite rare that ophthalmologists managed to publish in these journals. Why? Why did we do it? Because there were implications of looking into the eye, into the general health, looking at the brain health. And we were lucky enough to have also an editorial in that issue by the big guru, Isaac Kohane, the big guru of artificial intelligence at Harvard. And this was followed by many other papers. But in conclusion, I think just to remind you, I think it is very important for the younger generation. Choose your environment, choose your mentors, as Rachel told you. Choose your topic very carefully. I think it's not as important what are you studying, but who are you studying this with. Choose your target when it comes to publications. Choose your collaborators. If you think you are the smartest in the room, you are probably in the wrong room. And most importantly, choose your values. Why? Because why are we doing all this? For the patients. This is a picture that has been taken in Ho Chi Minh a couple of years ago, as I was coming to do surgeries. I'd like to thank you very much. Thank you very much.
Right, Dan, one question. Okay, so you can see Dan's, I think one thing that all of us will see is that as a neuro-ophthalmologist, he's very enthusiastic about his field. Okay, so Dan, why did you pick as a trainee, you know, many young people here, why did you pick neuro-ophthalmology as a career path? Because, you know, when people are looking at careers, many of them want to do cataract, many of them want to do refractive, look at VR, and the number of neuro-ophthalmologists generally, I have to say, is a bit more of an unfancied subspecialty. Why did you pick neuro-ophthalmology?
It's a great question. So the reason is, I think I initially, I mean, there's what, come on, you must agree with me. There is nothing more fascinating in the world than the brain. And I wanted to be a neurologist at the beginning, but I thought I cannot do much for those patients. That was 20 something years back. And then I thought, well, the eyes, I keep telling the neurologist today that the brain is just an expansion of the eye, although it should be the other way around. So, and I was just fascinated by the eye, and there was an obvious choice because those two fields are so much integrated, and it's wonderful to be able to be a doctor. I am a doctor because I'm taking care of patients with. We just saw a patient with GPA, with Wegener, a horrible condition, and Friday. It's complicated. I feel like I'm a doctor. When you choose to be a doctor, you didn't want to do capsulorhexis. You wanted to help people. And that's what I'm trying to do. And I think neuro-ophthalmology is one of the windows to that field.
Great, thanks, Dan. That's excellent. Great. Okay, so I'd like to invite my next speaker, who's Dr. Katrina Bell. She's another clinician scientist and looking at fundamental work in glaucoma. Thank you.
Yes, hello. I'll just wait for my slides. Okay, so at first I'd like to thank the chair of the session, Prof. Mehta, for giving me the opportunity to talk here about how my research or how research helps my clinical practice. So just a little bit about myself. I am from a small town in Germany called Mainz. It's probably well known for Johannes Gutenberg, who developed the mobile printing press in 1450, and within Germany, we are also well known for our carnival. On a more serious note, recently you may have heard of BioNTech. This is a company that was formed in Mainz whilst I was studying medicine by the clinician scientist Ugo Sahin. I was actually very fortunate that he was one of my teachers in medical school. Also, you surely have heard of Katalin Karikó, who is the lead scientist in BioNTech, and I also met her during my medical studies and had a small collaboration with her. So two really inspiring people that even if I feel that sometimes doing clinical or being clinician scientist is difficult, they inspire me to continue. So why do I do research? I've always loved biology and learning more about how all the processes that underlie diseases. And I wanted to use this passion to help patients to find new treatment strategies, and to find new treatment strategies and options for optic nerve diseases. So I figured if I want to do this, I would have to study medicine in order to be able to address clinical questions with research. But as we know, there's quite a large gap between the lab and the clinic. And the way there is often quite foggy, and only standing on one side, it's very difficult to see the other side. So in order to be a clinician scientist, I already started fairly early to also do some basic research during my medical school studies in Mainz, which I then continued during my ophthalmology training, which I also did in Mainz. This quite often meant doing weekends or evening shifts in the lab because we actually do not have a clinician scientist path in Germany. It, however, allowed me to complete my PhD looking at the role of neuroprotection in retinal ganglion cells in cell culture models, but also animal models. So after I had finished my clinical training, I was wondering, what's the next step? How can I possibly make a difference and improve the outcome for the patients I am seeing? For this, I got interested in the question, why does aging predispose to most blinding eye diseases? And more particularly, in my case, why does aging drive glaucoma? As we all know, the prevalence of glaucoma increases with increasing age exponentially. And there are many factors that could play a role for why does aging drive glaucoma, such as bioenergetic failure, neuroinflammation, or vascular dysregulation. At this time point, I got very interested in looking at autophagy because this was the new kid on the block in the neuroscience, let's say, and had recently just gained a interest because it had been rewarded a Nobel Prize in 2016. So I thought, so why autophagy? Also, it's a little bit about autophagy. It is a major modifier of aging, and it allows the recycling of organelles, a bit like our trash recycling mechanisms. And we, at this time, knew that autophagy decreases in the retina with age. Most excitingly, studies had recently shown that there are several autophagy genes, such as the optineurin and the TPK1, which are associated with an increased risk of glaucoma. And I'm currently studying some of the effects of these gene mutations. So as a young clinician scientist, this was a super interesting field for me, which offered a lot of opportunities to look at aging and glaucoma. So I thought, how can I best do this? I felt that after the clinical training, I needed to get a better understanding of basic lab research, although I had already done my PhD. So I decided to do a postdoc and looked around the world to see who is the best person I can do this with. And I found Patricia Boyer in Madrid, who's a world-leading expert in autophagy in the retina. So I applied for a scholarship in the German Research Council, which I was lucky to get, and I could spend 2 years in Madrid. Here I could focus on 100% bench research, and I was working in a lab conducting very high quality research. I also found a lot of new collaborators, and we've heard in the past talks how important this is. This opened up a whole new research field for me, and I was able to publish papers in this new field. The cons were that I did actually for these 2 years have to take a massive pay cut. Although this was a super prestigious scholarship, and I couldn't actually do clinical work for these 2 years. Above that, I actually couldn't speak any Spanish, so it was quite difficult to get around. Anyhow, during this time and after my postdoc, I sort of thought, what have I learned so far? I had learned it's not really very easy. And especially if you stand only on one side, the basic research side, or only on the clinical side, it's almost impossible to stretch yourself to reach to the other side. I also had learned that experiments unfortunately don't always work out the way you expect. Even if you start and try and chase something, it might not be able to answer your question. I also found that actually being a clinician scientist, you need many more additional skills rather than only research. You have to be able to manage your time and build and manage teams, but also manage budgets, write successful grants, be able to write good publications, and be able to present your data well, and build and manage collaborations. And these are actually skills you can also use and need in your clinical practice as well. I also figured that it is very important to have a good work environment. And ideally, I thought I would want to work in a center of excellence that attracts excellent clinicians and researchers with a supportive chair, hospital and university, and also adequate facilities for conducting research, such as big clinics with appropriate patients and databases, good labs with good space and infrastructure, the ability to collaborate with clinicians and scientists within and outside the eye. But also, it needs to be a work environment that can provide you with the time and money you actually need to do to be a clinician scientist and do research. So I was very lucky that I was able to come to work in Singapore at SNEC in SERI in 2020. This is, as we've already heard, SNEC in SERI is one of the leading research institutes and eye hospitals, and it has a very strong drive for translational research. It also has a very clear clinician scientist path, and which allows me to have some protected research time, 70% of my time and 30% clinical work, along with strong mentorship. So I'd just like to show two examples of how Singapore has helped me so far. There are very good funding opportunities, which is super important if you want to be a clinician scientist. I was able to receive an NMRC New Investigator grant, which allowed me to look further at my field of interest, looking at autophagy and aging in glaucoma. I also benefited from strong mentorship from my mentor, Prof. Ong Tin, who gave me the opportunity to investigate the role of a gene mutation associated with an aggressive form of exfoliation glaucoma in a specific clinical cohort, a study we were able to publish in Ophthalmology. So all of this might seem like a really long and tedious journey, but I do feel that it's very worthwhile. And to put this into perspective, David Mackey, a Australian ophthalmologist, looked at big innovations that have happened in ophthalmology in the past. For example, the intraocular lens, but also anti-VEGF for wet AMD. And it took more than 30 years to translate anything from the first manuscript to the clinical practice. So sometimes you really need to be patient. So to sum it up, how does my lab and clinical work help each other? My training in science and clinics allows me to address clinical questions with the goal of improving clinical outcomes. It has also helped me to identify and now evaluate new therapeutic approaches. It additionally allows me to travel, meet and collaborate with some amazing clinicians and scientists around the world and build up my knowledge base. So personally, I do believe that being active in research has helped my clinical practice. Thank you very much.
Great, thanks, Katrina. Okay, so one thing that comes out obviously in your presentation is one key factor that I think that everyone as clinician scientists need to have is determination, right? I mean, you took a big gamble and went to Spain. I mean, like you said, I don't know, can you speak Spanish now?
Not that
No,
well.
okay, not that well, okay. So but you took a big determination. You left Germany and then you went to Spain, and then you've been up in basically in Singapore as well. And I think that, and also different to Rachel, where you did your PhD quite early because you even started doing it at medical school. So, I mean, that's very young, to be fair, when you're a medical student, to think that I'm going to be a clinician scientist when I'm a clinician. Most people have no idea what they don't even know what subspecialty they're going to go into. So yet alone to be a clinician scientist. So what was the trigger at that time period to give you that idea that I'm going to be a clinician scientist? And then why clinician scientists say in ophthalmology?
Okay, so I wanted to be a clinician scientist when I was in school. So I was thinking of, do I study biology or medicine? And I decided to study medicine because I thought it would be easier to translate into clinics and to actually understand the patients and what are the patient's needs. So at school, I already worked in a lab, and then it was logic for me to just continue working. And I was always interested in neurons. And I knocked on the door in ophthalmology and also neurology, and I happened to get a really good PhD subject in ophthalmology. And also the advantage then is actually ophthalmology is a good field to be in for research. It's actually, you can visualize it, you can see what you are examining, and rather than just doing an MRI or something like that. And they also gave me the opportunity to have, to pursue my research further.
Okay, thanks, Katrina. Okay, any questions from the floor? Yeah. Yeah, of course. The introduction.
A quick question, because I think it's very relevant to your talk, but this question is for the panel, and I think it concerns the younger generation. So everybody in the panel, if you had to redo everything, would you do research first or clinical stuff first? Rachel?
Yeah, I think that's a great question. I think for me, I would probably still go the same route. I mean, I feel like which is clinics first and then exploring research, because for me, I feel like it was very important to be able to frame my scientific research interest in terms of where things were going clinically.
I would probably still stick to doing basic research first, just because it's my passion. And it takes quite a long time to learn, actually, and to learn different models, understand what the pros and cons, and read enough literature to find the focus you can, or the question you can focus on and hopefully answer.
Professor Mehta?
Sitting on the fence, right? So it tells you you can succeed in either way, right? That's the message I've taken from both of them. I mean, I think Kat's quite special in the fact that she had the determination at a young age, and the gamble to do that is that, of course, you may end up doing research in a field that you don't subspecialize in when you become a clinician. Well, if you do it later on, like I did as well, same as Rachel, then your clinical, you have an idea, okay, I'm going to be a corneal surgeon, I'm going to be a glaucoma surgeon, and then you focus on your research, and then goes down, and you go into detail on there itself. But I think that, it just shows you that depending on different people's personality, you can do well, either way that you choose to do it.
Thank you.
Great, thanks, Ken. Okay, so I'll do the last talk then. So I'm going to give you a little bit of introduction about Seri, and can I have my slides, please? Okay, so I have to look after all these people and find ways of keeping them funded and keeping them busy as well. So obviously, Seri is the Singapore Eye Research Institute. And how did it basically all start? So Seri was basically, it's a national body for ophthalmic and vision research in Singapore. It was initially founded in 1997 and funded by the government. And we basically survive on competitive grants from different facilities within Singapore and also basically outside Singapore as well. And it's really the research arm of SNEC, as you've seen from the pictures that lies across the road. Initially, we were affiliated with NUS, but now we're basically with Duke NUS itself. The idea is, or the mission statement is, to really conduct innovative and world-leading basic science, clinical, and translational eye and vision research. Okay, and the key is really the translation eye and vision research part, and I'll show you how we basically gauge this from the work that we basically do. So our founding father is basically Professor Arthur Lim, who's quite a well-known figure in Asia. And the idea, or the idea was to build a national center of the highest quality clinical care, which obviously is SNEC, but with the most innovative methods, but cutting edge research, because the way of basically maintaining our situation as a clinical center is really through our research, or research basically innovation as well. This is where we started back in 2001. So Seri was all over the place. There was one floor in the main building, which was the lab, which you can see on level 6. And we have a clinical trials unit in level 5. I came to Singapore in 2007, 2006. And you can see that list of the labs on level 6. And I remember we used to go for lunch, and basically the whole of Seri lab team could fit on basically one table for lunch. It was actually that small. Over the years, it's basically developed, and this is basically where we are now, where we have 2 floors in that building. You can see on the right-hand side, we have 1 floor in the for office and dry lab space, which is level 6, and we have a whole suite of animal facility downstairs that's underground in the basement, where we have a whole ophthalmic suite for animal research, all the way from small rodents all the way up to NHPs as well, non-human primates. And really, we're lucky in the fact that what we see is a partnership between our clinical service, which is SNEC, research side, which is CERI, and our academic side, which is Duke and US. And you'll see all our affiliations are linked to Duke University in the United States. What was the aim? The aim was basically to be one of the top 5 research institutes, not just in Asia Pacific, and basically in the world. And that was our real basically goal itself. So how do we basically develop this from scratch, or as a template itself? And the idea is that we were basically going to always focus on translational clinical research, okay? But we had to think of long-term strategies, obviously financially. You've got to have, as you've seen from the speakers, the interaction between clinicians to come up with those questions, and really clinician scientists to make that bridge to the basic scientific community itself. And we wanted to basically have a broad impact on all aspects of ophthalmology. There's no point doing the research, publishing your paper, presenting at a meeting. You've got to make that big impact into the actual patients as well. And this is a sort of this virtual cycle of growth that we basically look at. We have an innovative idea, you basically perform the research, you try to basically improve the outcomes, and then you try to implement that, you try to get grants for if it's basically successful, and then basically put that back into the research as well. And you have this innovative cycle of growth. And of course, it's difficult to basically achieve all these things, but if it's possible, then you can see how this basically cycle can be replicable basically going forwards. Now, over the years since the founding father, we've had several executive directors. So I took over from Tin at the end in January 2022, when I was looking a lot younger than I do now in real life. And you can see that initially we basically had 5 translational clinical platforms, and this was to support our research. So you can see we had a basic laboratory research side, we had a preclinical animal platform, because we realized that if we're going to translate our work, we must have a good preclinical animal facility, and we have amazing equipment. We have 1 femtosecond laser, we have Excimer laser, we have intraoperative OCT, we have a whole ERG suite, imaging, fluorescent angiography, all for animals. And it's important to be able to have that if we're going to basically translate our work. The next phase of translation, obviously the clinical trials unit, and then we have a population health unit, and then of course our technology and development. And these are the guys that take our innovations to companies that hopefully they can then be licensed out and commercialized internationally. And I'll show you some examples of that later on. CERI's grown. Okay, when I came, CERI was basically 100 people. Now we have to look after almost 250 people. So people told me, what do I think about when I wake up in the morning? And I think about how I'm going to feed 253 people, because I have to think about how much we're going to basically get money into CERI to keep everybody's salary and keep them all alive. So how do we measure the success? I mean, how do we know whether we've been successful since the 1997? Well, if you look at the number of publications, this is just from 2008 to 2021. You can see in 2008, we were publishing 161 papers a year. Last year, I mean, despite COVID, we published almost 460 publications. And this is just from one institution, just our institution. And if you look at the bars, they basically divide up into dark blue to light blue. So it shows you the level or the quality of the science with respect to impact factors. So the largest blue bar, the highest impact factor journals as well. So not only has our volume increased in what we're producing, it's also basically the quality has basically improved over that time period. But the focus really is not just to be the best in Southeast Asia, it's really to be competing on an international scale. And the way we get that is other metrics. So this is from a US website called expertscape.com, where they basically look at experts in the field of many of the subspecialties that we see in ophthalmology. And you can see many of our faculty are ranked either in the top 1 to 4 or top 5 in the world in many of the common diseases, in cornea, angle closure glaucoma, retinal diseases, deep learning, glaucoma, cataract surgery, myopia, and also in refractive surgical procedures. And this is an objective way of a measurement of how well we're basically doing as well. Likewise, Excelvia Stanford did a survey recently, and they looked at basically the top 2% of everybody basically in the field itself. And there were 21 scientists from CERI and SNEC who identified as the top 2% of people who published in basically our field and basically ophthalmology. And some of these you may recognize from speaking today as well. And of course, there are other sort of less scientific measures as well, such as things like this from this power list, where several of our faculty were basically also recognized as well. But the other way has been basically success breeds success, and you'll always hear that basically over the time. And of course, as we become more innovative and produce more papers, you can see our annual budget has gone up from 12 million back in 2008, 2009, to now to a running budget of almost 44 million. Okay, and that's how much we need to basically keep on bringing in every year to keep our faculty basically going, to be able to produce this level of research. And we've been lucky in that way because we've been able to link that to our publication success. But one of the key things that I have to show you is really this, and this is basically the innovation. Okay, and we were the first hospital in Singapore to have a proper technology development industries, and all these things you'll see on the right are companies that are examples of licensed technologies that have been developed in SERI, SNEC, and then licensed to companies, or these are startup companies that have been come out from research from our research institute. Okay, so whether this is basically Plana, which is this app for myopia, they almost have 500,000 people on their database for basically myopia research itself. EndoGlide is a device that I developed, which is basically for corneal transplantation. Spiralis is a device used for basic cataract surgery to basically open up pupil expander, which has basically been licensed to basically goiter. But these are all innovative works. And of course, you see, when you basically produce something in the lab, ideally you want to treat your own patients that you see in your clinic. But if you can treat patients miles away around the world, in Europe, United States, South America, with the technology that you develop in your own research institute, of course, your impact is huge. Okay, and of course, going along with this, we work with several companies in order to aid us, to help us to be able to develop these technologies to basically take them into market. And this is our technology domain now. Okay, so these are all these things you'll see over here in therapeutics, devices, diagnosis, softwares that have been basically developed in the research institute. So you see Dan's Bonsai portfolio over here on the right-hand side. And all of these ones with the stars on them itself are things that have been licensed, commercialized to the company. So the inventor who developed it in SNEC or SERI will receive a licensing agreement and receive money every time that instrumentation is basically used. Some of it will go back to the research institute, some of it will actually go back to the inventor. But this is really making a difference back to the patients, which is the ultimate goal, as you heard from basically all our research as well. And if you want to basically look in metrics, this is the overall metrics of figures over basically the time period since 1997. So almost 4,500 basically publications. We have 23 clinician scientists and 385 million competitive grant funding that we basically won over that time period. And this is competitive grants. These are not grants that basically you have to compete to basically get these grants yourself. But again, success breeds success. It allows you to basically do things, and then you can actually develop your research focus and your effect on translation as well. So this is a current platform basically for SERI. So myself as executive director, we have basically 6 directors in different fields, from population health, translational research, and administration and laboratory and translation research and clinical translation research. We have 16 research groups supported basically by 7 basically research platforms. And now we have joint labs. So we have joint labs with the local universities in engineering. So we're looking at cross-fertilization of ideas, but also with industry as well, with JJV and also basically with Santen, where they have joint labs in our research institute as well. So in summary, it is important to maintain a key initiative of impactful translational research, because at the end of the day, you've had more speakers. What we're trying to do is improve the outcomes of our patients. Okay, and we looked at realignments. We keep on evolving over the years. We look at new areas of research such as AI, aging eyes as well, as basically things have evolved and technologies basically evolved around as well. Definitely with the ecosystem, you heard how AI has become a big area to basically focus on. You need to keep on evolving. We look at a hybridization of fertilization of ideas with engineers, with joint labs, with industry like IHPC, which is a high performance computing unit from A-Star, relating it back to basic science, and also basically joint lab to an industry as well. And it's important to identify, obviously, the next generation. We want to keep this going in the future as well. And that's why it's important to bring some younger people than me to give you talks as well today, and not just basically the full professors that are coming to give talks. And the idea is that we can keep this research innovation going, because of course we want the next generation to sustain this for the future as well. Thank you for your attention. We can take any questions from anyone? For any other panelists? Yeah, Dan.
Excuse me. I understand, and I have a question for you. From your experience, and in Vietnam, we don't have a lot of big hospital. So could you give some advice to balance between research and financial arrangement for small hospital in Vietnam?
Yeah. So the question is basically balancing research and financial. Say again?
Could you give some advice
Yeah.
for the hospital in Vietnam balance between research direction and
Yeah.
business direction?
Yeah. So,
Yeah.
I mean, so you have to take, I mean, you'll see that picture I showed you of all the executive directors who have then gone on to be directors of our clinical unit. It's because it has to come from the top. So the person in charge has to realize the value of research and why it's basically important. And it's not a short journey, as Katrina showed. It can take years for some of this revenue and some of this thing to come back into basically clinical practice, or even inventions, even devices to come back and affect your basically clinical practice. And researchers may not be for everybody, but everybody questions that patient that they see in clinic and they wonder, can we do something better? And if you ever think that, if you ever see a patient in your clinical practice and you think, God, this is terrible, can I do something better? Okay, that's your spark of your research question. Now, the next question is whether you have the ability to find people around you who can help you answer that question. And the thing is, the same as SNEC. That's why I showed that picture of the staffing level. You know, when it started in 1997, SERI had 11 people. Now we have 250. Now, we didn't jump to 250. When people come and visit SERI, they're, oh my God, we're never going to be able to do this. But you have to realize that we started from very small numbers. So you start off small, okay, and you have to keep on building and try to build up on success. So once you get a few small hits with successful things itself, you can then build that up. Not everybody will take time out, like everybody on this panel, to go and do a PhD, but they may take time out to go and do some research, maybe a master's degree in basic research itself. And you identify some of the people who basically want to do it. Often it's slightly younger generation who are inquisitive and they want to basically try to do something different. And really the goal is that in the big institutions here, you have tons of AI. I used to go to Hanoi as well, to the NIO, and also basically to Ho Chi Minh as well, to the hospital there to do surgery and stuff. Well, you have a lot of patients. It's difficult to do research when there's a lot of patients, but it requires a bit of organization and you have to give people time. Okay, if you're having to see 34, 40 patients in a clinic and then having to do a research project, it's not going to work like that. Okay, you need to then identify the right people and give them protective time and get them the right training and then see what they can basically do. And I think that, this shows you from what I showed that it's possible, right? With the right encouragement and the right environment. And my job now as director is to give these guys the right environment to produce, right? But if you told me that, do you think it's possible to produce 460 papers in a year? Of course, I tell you that's impossible. But but you can see from the chart, this is what was being produced at our institution. So it is doable. It's not something that I would necessarily aspire to every year to be able to do, but it just shows you it's possible.
There's no one. Can I ask a question, please? Right. So there are a lot of young people in the room. So I was wondering, this is a regional meeting. What can SERI offer to young guys who aspire to do research or want to collaborate with us?
Yeah, so that's a good question, Dan. So basically, we do offer like short-term scholarships, okay, and also observerships and stuff where people can come and work with the teams that we have on the platforms that I showed you, basically in SERI as well, and also basically in SNEC. So I get a lot of fellows from Europe, from Korea as well, and Australia to come and do time period. But if people want to come, if they want to pick an area like AI, for example, or even laboratory-based research in one of the areas that they do, they work on that ops chart, like I showed you, is basically on our website. So if you look at the website, you can contact the PI. There are funds available as well to do some sponsorship as well for when they come, and they can come for a period of basically any time, basically from 1 month up to 3 months. Then when they come and do an observership, maybe do a small project during that time period to learn some skills. So this is available for everybody to come, a possibility to come to as well. If they want to mix it with clinical, also that's possible as well. Okay.
I have a question. I'm Dr. Rajat. I'm from India, and this question will be relevant even for our Vietnamese friends also. So, see, there are established centers across the world like SNEC or for that matter, LV Prasad in India, where there's a very environment is very research based. Even the clinicians are like going ahead with some kind of clinical research, and there is a translation component to it as well. But the challenge lies when you are part of smaller institutes where you don't really necessarily have the infrastructure to kind of, you may have ideas, but you do not have the infrastructure to kind of take it through. And the problem happens is when you try to pitch the same for, some kind of grants or some kind of, so if there is no previous history of like, running through these grants successfully and completing projects, the sponsors are usually not very happy to kind of oblige for your requests. So how do you think we can,
That's right. So one of the things you saw on everybody's slides was collaborate. Right? Collaboration is the key, right? And I mean, you know, when you're giving out money, or when we're giving out money in grants, $1.5 million, $2 million, right? It's like going to a bookie and looking at a racehorse, right? You look at a racehorse. If I'm in India and I look at the racehorse, obviously I think, okay, if I go to LVP, I go to Ames, I go to Arvind, I go to Shankar, there's a good chance they're going to be able to do the work. If I go to an institute I've never heard of, never heard of the people, they have no publication record, I'm not going to give them $1 million. Right? And that's the answer. So you basically collaborate, you get people around you. None of the staff, none of us who basically on any of those pictures that I showed you are there because they work by themselves. So we have a team, we work together. Team research is not a one-man pony. Okay, cataract surgery is a one-man port. You do your surgery, you leave OT, you tap yourself on the back. Thanks very much, I've done the surgery. Research is not a one-man team. Okay, it's always a group around you, and what you achieve as a group is, you know, when you get your publication out and stuff, it's always a team. That's why you see multiple authors that are contributing. So that's my advice would be collaborate. There's very good big centers in India where people can easily collaborate. And if you're in a smaller unit, you focus on one thing. You cannot obviously do everything like we do, or I showed in the pictures. You focus on something that you see and you think this is the important answer. You see the patients, you know, you see something that you want to change for that person sitting in front of you. That's your basically initial teaser for what your clinical improvement needs to be, and then you take it from there. Okay. Yep. Okay, great. Thank you very much, everyone. Thank you for coming. Thank you.